Hello, in this video we're going to be looking at the electron transport chain and chemiosmosis part 2. So let's not waste any of your time. Let's get right to it. Now, part 2, electron transport chain, chemiosmosis. Let's remind ourselves where we've been in cytoplasm for glycolysis, inside that inner membrane for pyruvate oxidation, into the middle of the big area of the mitochondria. And now we're at that membrane on the far side, and you can see the proteins there, and the idea of when each of the electron carriers drop off their H's and they move through, now they're in that outer membrane, as I mentioned, up against the membrane of the edge, and they move back into the middle area, going through ATP synthase and making those ATP molecules. Now, when we think about this process, we certainly want to think about, kind of the assembly line manner of this. And this diagram here, this obviously is a water wheel. And if you know anything about history, like sawmills and the early days, especially prior to electricity, one of the ways that you could have a plant like a sawmill where the saw could be, ran not by human hands, and then you could just run lumber through there, is to use a water wheel to generate the power to operate a machine. Steamboats, for example, were the early process of this. The engine generated the steam, it turned the wheel, which moved the boat through the water. But when you think electron transport chain and chemiosmosis, we've got the idea of those pumps, water moving along. We're dropping off the H's, and then the water's flowing through that area and carrying all these H's to the water wheel and down and around that, and that ATP synthase, and then making those ATP molecules. So really think about it as far as an assembly line and a production line to make these products. Alright, so here's our summary slide for the electron transport chain and chemiosmosis. The idea now is that the end products are obviously a little different. One, we have a huge number of ATP molecules, 32, and the idea of water being made at that protein number 3, where the oxygen accepts some of those electrons, and of course the difference in a different location here is the fact that now some of our end products are those electron carriers, because when they become oxidized, they become listed in the end products. Instead of, now they no longer have the H's, but end products nonetheless. And of course, it doesn't have a number there, but obviously it could be, it should be a number for each one of them that were dropped off. So we know those numbers were 10 and 4. 10 and 2, I'm sorry. But just remember, NADH drops off at that first protein, FADH drops off at that second protein, and then oxygen takes H's at that third protein. And of course, all the H's work their way down through their membrane, their protein, and then across, the ATP synthase, and work their way through that ATP synthase, that specialized pump, to generate the ATP. And of course, the electrons on the H's, that's the ones that activate those active transport pumps, as I mentioned. Active because we're taking materials that we have some of and moving them into areas where we have a bunch. So that's the active transport part of it. Now here's the slide 44 that I've been talking about. This has them all 4, where you can easily see them. Locations, starting materials, end products. So this is the slide. If you could only take one thing into battle when it comes to the test, this would be the one thing that you want to grab a hold to, and really the best thing to do is just to memorize it, and to where you can repeat it back and forth, frontwards, backwards, whatever. That there isn't any part of this that someone would ask you verbally and you could not answer it. That's the best process. If you can do that, then you will answer majority of the questions without any difficulty right off the bat. So here's slide 44. Be sure you have it well under control when it comes test time. Alright, so these next couple of slides and screens, we're going to talk about these big yield numbers. Now, everywhere, if you look around at different textbooks and websites and whatever, you will see different numbers for the net result or the grand total of ATP production. Excuse me. And there's some good reasons for that. But you'll see anywhere from 36 to 38, actual energy yields 30 to 32 for eukaryotes and 32 to 34 for prokaryotes. But there's a reason for that. That reduced yield, sometimes the membrane lets things slip through and they don't go through the proteins like they need to. But just things happen to where an HO2 will get away, and so not all the ATP that is could have been made is made. It's just like with your DNA. There are things in place to make sure that there's no mistakes when they're pairing the nucleotides together. But mistakes happen, mutations occur because of that, and things like that. So it is what it is. So that based on the leakiness of those H's will reduce the number of ATP molecules that are made, because if you don't have if you lose some of your H's, then you can't make as many as you could have. It's just like if you're making an apple pie and you got your apples there and you're just going to make a huge pie. It's going to be full, but then somebody comes along and sees the apples and grabs one and goes eat it, or they put it in their lunch for later, and you come back and you're missing an apple or two. You're still going to make apple pie. Okay, you're still going to make a pretty good one, but it won't be nearly as big or full as it could have been with all of the H's. Alright, so here's a diagram of ATP molecules that are made at each level. Glycolysis, making a couple, and NADH is carrying it, carrying them to the chemiosmosis, the ETC, the Krebs-Solve mechanism. So we come down to a net yield of 32. We make 34, but we use 2 in the initial part of glycolysis to get that going, so for a net of 32. So what we'll establish, since we just identified the fact that book to book, website to website, the number could be a little differently, that we will set the number as 32, okay, for our grand net total of ATP. So if there's any test question and it's asking for that net total, 32 is the number that we'll just agree on, that we'll look for in a test. Now, if the question asks for a section, like in glycolysis, how many ATP are made, then we're looking for that number. Or in the Krebs cycle, how many ATP molecules are made. Then you'll need their own particular number. But if it doesn't mention a section or a step, it wants the grand total number, and that number will be 32. Well, that's our video for this time. When we come back together, we're now going to be looking at a respiration without oxygen. So we'll see you then.